No, no, tranquillo.
We are ready now to start the session that deals with research projects supported by Parent Project. A short introduction, and then I will give the floor to Gloria, who is the other chairperson. We have never given up. Funding the research has been one of our objectives. This is something we have been doing over the last 25 years with the same determination. We have realized that the funds to finance research on a rare disease such as ours would always have been very few. It is a rare diseases, and researchers that have ideas and want to start projects spend 70% of their time to look for funds rather than carrying out research. Research projects supported by parent projects are the result of our determination at concentrating on research, great commitment of the families in fundraising, but also in acquiring new knowledge ourselves. We need time, a lot of time. You must overcome a variety of difficulties, and it is important never to give up. Funding basic research, that's a research that studies the mechanism enabling one to understand how the pathology works, enables to open up new way out, nothing immediate or very close, but something that is essential to reach in the future some therapies. We must try and understand the mechanisms of the disease and fight its progression. And one day, well, we hope that the dream will come true, finding a cure. And for some people, well, we should have funded only the most advanced stages of research, but deciding to fund the different stages in the development of a drug has led us at very satisfactory results. Think about Givinostat. We started from the very beginning, from the idea to the drug development. Isn't that a great goal? We have always been the protagonist in choosing projects. We have always trusted science, but we have always stayed within the decision-making process. Over these 25 years, we have never stopped following the projects, monitoring results, and trying to find a therapy that still is not there. We have exerted pressure, as Dr. Tuccerone says. We have tried to understand what has worked and what hasn't, what we could ask to whom, and trying to understand how drugs worked and better stratify the population, implementing and improving data collection, and understanding how necessary it is to have international cooperation. Supporting research for parent project means, as meant and means, having a dialogue with researchers, centers, all stakeholders, and being, as patients and families, also the protagonists of this dialogue. Well, this event is clear evidence of all that. And now the floor to Gloria, and I wish to welcome our rapporteurs and thank you for being here. Thank you, Maria Rosaria. Thanks you all for the attention. Good evening. Well, for me, it is a great honor to chair this session and introducing the activity of the science area. You'll be meeting other members of the group. We have three biologists, myself, Fernanda De Angelis and Ilaria Zito, and we manage all the activities having to do with research support and communication, project selection, and also patient register. We also provide consultancy service to the families who have DMD or BMD children. Well, of course, in the past, we had meetings a bit all over Italy, but unfortunately, this pandemic has stopped that activity, but we'll be resuming it as soon as possible. But one of the essential activities of the association is the support to research. It is a mission, it is an objection that we have been developing over the years, especially since 2005. We have funded research projects for a value that I had to read and reread to believe it, 3,829,000 some euros. And this is an incredible figure if if you think that this money has been collected by a patients association, parents association, and devoted to research. The projects we are introducing this evening started between 2016 and the first months of 2018. They are so they've almost been concluded. We have a few others that have started later, and as Maria Rosaria said, we wanted to fund basic research, basic science, so the initial stages of the pathologies, and then also applied research. The projects have already produced a number of results, high-level scientific papers, and of course, all that makes us hope in a future of clinical application, of clinical use. Over the years, the different proposals were being examined first by the scientific bureau and then by the parents who follow the science area, and then gradually they were sent to external auditors, as is the case all over the world for scientific research. When I started working in Parent Project in 2018, together with all the members of the staff and of the steering committee, we launched a new call, and this call included this idea of peer review. And so the projects, after an initial selection by our scientific bureau, well, these projects then were sent to external auditors, international external auditors, in order to guarantee absolute transparency and an appropriate auditing of the funding process itself, funding of research, which is a very important feature, as something we firmly believe in. I'm extremely glad this evening to introduce these 5 research projects. They are, most of them have already been completed, and I want now to give the floor to the different researchers, and more specifically now to Dr. Chiappalupi from the Perugia University. And he'll be speaking about a project that has been developed by Professor Sorci on the use of very special cells, micro encapsulated circuitry cells for DMD. This project went on for 2 years. It started in March 2017, and then with a small extension, and then it was concluded in August 2019. Thank you, Sara. You have the floor.
Grazie, Gloria.
Thank you, Gloria.
Buonasera a tutti.
So, good evening.
Si vede bene, sì?
I hope you can hear the screen well.
Le cellule di sertoli si trovano normalmente nel
sertoli
testicono,
cells
sono la componente più abbondante di tubuli seminiferi e sono indispensabili affinché avvenga la spermatogenesi. E sì, infatti si
of paramount
cernano numerosi
importance
fattori
for
trofici ed immunomodulatori che
spermatogenesis,
permettono la
and
protezione
they are
e
actually
lo sviluppo
responsible
delle
for
cellule
several
germinali. Grazie a
immunomodulatory
queste loro
aspects,
peculiari
thanks
caratteristiche, le
to
cellule di
which
Sertoli sono state utilizzate
they
con
have
successo
been
in
used
numerosi
and
approcci
successfully so in
sperimentali
the
preclinici,
framework of
come il diabete
different
tipo 1, la
investigational
malattia di Huntington, la
approaches
sindrome di
for, say,
Laron e la
Huntington
distrofia
disease
muscolare di
or
Duchenne. In
DM1, but
particolare alcuni
also
anni fa abbiamo
DMD.
messo a punto un nuovo
Actually, a
approccio
few years ago, we
sperimentale per la distrofia
implemented a
muscolare
new
di Duchenne
investigational
basato sull'utilizzo di cellule di
approach
Sertoli,
based
in
on the use
collaborazione
of
con il
such
gruppo del professor Giovanni
such
Luca e del
certainly
professor
cells as
Calafiore che da
sex.
anni si
We
occupano
did
di
that in
microincapsulamento
cooperation
delle
with
cellule
other
di Sertoli.
researchers,
In
including Professor
particolare, cellule
Calafiore,
di
who's been dealing
Sertoli
with
porcine sono
such a
state microincapsulate in
microencapsulated sex
microcapsule di
for quite
alginato
some
altamente
time. So,
biocompatibili e inoculate nella
biocompatible
cavità peritoneale di
arginate
topi distrofici di 4 settimane nella fase acuta della patologia.
into the mice. The speaker is going very very fast, actually. Maybe she might want to speak less fast than that. So anyway, inflammatory factors, and then from the peritoneal cavity, they may reach out to other organs, thus leading to reduction of necrosis, fibrosis, and adipose tissue deposition. And for the first time, we have also seen that such sex release heregulin beta 1 that may regulate a substance that may actually be of some relevance. All these factors work together, and they give rise to a better muscular activity. So, we have also considered this in view of a possible future implementation. So sex were injected into myotubes derivated from dystrophic mice, and we saw that sex can actually improve the situation independently of the mutation, of the dystrophin mutation of relevance, and we have also investigated the dosage, and we have seen that even a 10 factor lower dose may give rise to a significant reduction of the muscular necrosis. And we have also carried out this treatment within other kinds of mice where eutrophin regulation cannot occur. So this mouse is much more similar to humans, and a 3-week treatment with MC6 actually is translated into a significant reduction of the muscular necrosis and of the inflammation in all of the muscles involved. So actually this is independent of the eutrophin regulation. These are two independent events. And we have also wondered whether sex could have an immunosuppression role, or on the other hand, immunomodulatory role, because this may actually be the case in spite of the absence of relevant data. So we have used two models here. One with Aspergillus fumigatus. It's quite an ordinary kind of mushroom in order to induce a pulmonary infection in immuno-impaired immunosuppressed subjects. So we injected this MC6, we infected the mice with this particular kind of fungus, and after 7 days of treatment, we saw that sex showed a significant reduction in pulmonary infections, and basically a total absence of the fungus within the lungs, which shows that sex definitely give rise to a good response, also thanks to a release of factors precisely able to produce such improvements. And then wild type mice were also treated. There was LLC and there was a melanoma in these mice, but in both cases we saw that after treatments with sex, the onset of the cancer was not enhanced. So there's no immunosuppression here, which means that the factors enhancing the presence of the tumor are simply absent here. There was also reduction in lung metastasis and no loss of body weight, differently from the mice with empty capsules being injected, which actually showed such loss in body weight. So sex could protect from cancer and from muscular diseases. This is obvious here. As you can see, the final result is very similar to the one we obtain with mice untreated. On the other hand, the control mice showed a higher score of loss in body weight. This is also confirmed by myosin that is typical of these mice, and there is also an inhibition of the genes supposedly leading to muscular degradation. Well, actually, this is apparently not the case when sex are injected. These data were also confirmed on an in vitro basis through the culture of sex with such myotubes. Well, actually, there are pro-inflammatory cytokines, though here in particular, dexamethasone may induce a significant reduction in the general conditions that is totally reversed by the presence of sex. Now, of course, there's a dose-dependent approach that should be considered. So in conclusion, such MC sex induce an improvement of symptoms in dystrophic mice. They induce the utrophin upregulation thanks to the beta-1 release in such a dystrophic mice. They reduce muscular infection in both kinds of mice that were tested, and safety is apparently there even 1 year further to the injection. Sex are still vital again after 1 year. And there is no immunosuppression, but on the other hand, there's an immunomodulation and sex releasing factors. Well, the factors released by sex are apparently protective regarding cancer. I'd like to thank all those who have allowed us to carry out this research. Here are the names of the people who should be acknowledged for that. And of course, I'd like to thank Parent Project. Thank you very much. Thank you.
Thank you, Sara, also for respecting time limits, and then we'll have time at the end for questions. And please write the questions on the chat, and then we'll read them at the end. And now we give the floor to Professor Maria Sofia Falzarano from Ferrara University. She's going to share with us a project that we enormously believed in. We had funded the first study for her group, and she'll be speaking about it. It is a biobank of stem cells of USC. So it It is a project that lasted for 3 years. It started in March 2018, and so it was a research project, applied research project. Well, thank you for being here, and you have the floor. Thank you. Thank you for the invitation. I'll be showing you an essential part of the studies we have carried out on urinary stem cells, on a focus on their use in diagnosis and research. USCs are an interesting cell model, and they can be obtained with simple and non-invasive techniques, non-painful techniques, so they can be repeated over time, and we can obtain a cell model that is very easy to use on the patient. Many studies have characterized this new model to study various diseases and genetic diseases, and we concentrated on the study of DMD. As Gloria just said, the first project that Parent Project funded enabled us to characterize dystrophin in urinary stem cells, both in controls and DMD patients. And so analysis of RNA in primary stem cells and in urinary stem cells, and also the expression of the dystrophin protein, and obtaining a result whereby this model can be there is a sound problem. The sound of the speaker comes and goes. It is impossible to make sense of what she's saying. I'm sorry.
fatto questa analisi in stem urinari di controllo, in stem urinari di DMD e in stem urinari di DMD trattate con una molecola antisenso per lo skipping esonico. E cosa abbiamo visto? Che, come succede per il muscolo, molti set di geni cosa che avevamo già valutato e abbiamo visto che risultava efficace. Venivano poi corretti dall'utilizzo di una molecola come l'antisenso e abbiamo visto che in effetti il trattamento portava a una correzione di molte di queste vie deregolate. Per quanto invece riguarda la caratterizzazione delle colture cellulari, le cellule staminali urinarie presentano delle caratteristiche molto simili alle cellule mesenchimali staminali ottenute da altri tessuti. Come tali, in cultura presentano diversi sottotipi cellulari, però a tutt'oggi non è noto un marcatore specifico che distingua e selezioni i diversi tipi cellulari in cultura. Questa mutazione presente nei due pazienti. Per quanto invece riguarda la diagnostica, abbiamo utilizzato le stem urinarie per trovare, identificare delle mutazioni atipiche complesse nel gene di una mutazione intronica profonda nell'introne 10 che portava poi a un'alterazione dello splicing che determina l'assenza della proteina distrofina in questo paziente. Nel paziente invece 2, così come nel paziente 3 che vi mostrerò successivamente, in entrambi c'era assenza di distrofina a livello della biopsia muscolare e l'analisi del trascritto distrofina aveva mostrato l'assenza di amplificazione di una grande porzione del trascritto distrofina. Questo ci aveva quindi suggerito che probabilmente alla base di questa la mutazione alla base del fenotipo di questo paziente fosse un grande riarrangiamento e infatti attraverso whole genome sequencing abbiamo valutato e abbiamo trovato una grande inversione che coinvolge una gran parte del gene distrofina e anche una regione vicina non codificante, una regione molto grande di circa 10 megabasi. Nel paziente stessa cosa, assenza di distrofina nella biopsia muscolare e una grande regione del gene del trascritto distrofina assente. Il whole genome sequencing ci ha permesso di trovare anche in questo paziente una grande inversione di circa 15 megabasi che porta all'assenza poi della proteina nel paziente. Quindi concludendo, possiamo dire che l'analisi trascrizionale con l'RNA sequencing ci ha dimostrato che questo nuovo modello cellulare può essere utilizzato non solo per lo studio in vitro del trascritto DMD, ma anche per lo studio di altri geni deregolati in questa patologia e potrebbe essere utilizzato anche per screening di nuove molecole, di nuovi farmaci. In particolare stiamo valutando sia a livello trascrizionale che di espressione della proteina altri geni correlati a patologie neuromuscolari per vedere se è un modello utilizzabile anche per altre patologie. L'uso invece del selector ci ha permesso di identificare 3 diverse sottopopolazioni cellulari e questa informazione ci può essere utile perché un'ulteriore e più approfondita caratterizzazione di ogni singola sottopopolazione potrebbe aiutarci a capire come sfruttare la nostra cultura per diverse metodiche di laboratorio come riprogrammazione cellulare o per individuare marcatori di malattia. Infine, l'applicazione in diagnostica ci ha dimostrato che l'analisi del trascritto DMD nelle stem urinarie in associazione con altre metodiche come il whole genome sequencing ci ha permesso di identificare delle mutazioni rare e complesse nel gene DMD, quindi ci suggerisce che questo modello potrebbe essere di riferimento per identificare altri tipi di mutazioni che non sono ancora state trovate nel gene DMD. Ringrazio tutto il mio gruppo di ricerca, in particolare il Parent Project per averci permesso di effettuare questo studio su questo nuovo modello cellulare e il gruppo del professore Schiglian della Stem Cell e del professor Bicciato dell'Università di Modena per aver collaborato e per averci aiutati in questo progetto. Grazie mille.
Grazie, grazie.
Thank you. Thank you very much indeed, Dr. Falzarano. We have been trying to solve some technical problems, some sound problems. If you want to ask questions, write them on the chat, and so that we can work more rapidly in the Q&A session. And now I give the floor to Dr. Roberto Rizzi of the Institute of Biomedical Technologies of the National Research Council. He received funds and the project started in 2017 and it has been almost completed. It is a very passionate project, the main objective of which was studying in model mice the effect of givinostat on the heart. Thank you, Roberto, for being here with us, and I give you the floor.
Devi smutarti.
Switch on the microphone, please.
Ok, buonasera a tutti.
Good evening. The heart is an organ that is innervated by nervous fibers that regulate the heart. The cardiac muscle has the same striatic aspect of other muscles, but the fibers are interconnected and they form a sort of reticulum. If a single fiber is stimulated, the whole of the syncytium is stimulated. So in the heart, you don't speak of contraction of individual fibers, but of a contraction of this whole thing that is called syncytium. And the impulse is self-generated. And if a person is in a situation of anxiety or excitation or any other emotional condition, the number of impulses coming from the central nervous system is shown in the heart. And the same happens if in the heart there are congenital defects or mutations that may affect the cardiac muscles and also interact with the central nervous system. The heart of dystrophic patients shows a marked dilative cardiomyopathy, dilative cardiomyopathy and an excess of fibers on the heart walls. We have started this study on this phenotype for DMD using the animals that were introduced, MDX mice. And we have seen that the presence of the inflammatory infiltrate was felt at 10 months, was seen at 10 months. We examined animals at 3 months, 10 months to see also how it changed also in terms of muscles. From the first analysis, we identified that as far as inflammation is concerned, that is extremely marked in the more advanced age, we saw a sympathetic denervation, and this sympathetic denervation was correlated with a massive remodeling of the extracellular matrix, and it was correlated with this substance, the CSPG4. It is a proteoglycan 4, and proteoglycans have now an important role in heart remodeling, and now they are considered as diagnostic biomarkers, specifically for heart fibrosis. And it was demonstrated that the presence of CSPG4 inhibited sympathetic innervation. And the presence was also found in myofibers. And so we thought, is it possible to have an excess of CSG4 of these proteoglycans in the extracellular matrix, and that may cause a sort of denervation of the system? To do that, well, you see what happens here. We have fibroblasts, but all this co-localization, this closeness between denervation and inflammation infiltrate led us to manufacture this engineered cells, the CAR Ts, to find a co-culture system and to try and understand which cells did produce CSPG4 in greater quantities, and fibroblasts were not affected by the presence of this engineered CAR T that were supposed to attack CSPG, but only macrophages, and especially in the M2 phenotypes, they were killed by these cells. This hypothesis was validated with a number of experiments, protein identification, and also a number of experiments of immunofluorescence. When with antibodies, we try to find proteins And so this hypothesis was confirmed. Then we also examined CHST11 that induces the production of CSPG4. Here you see a pattern, a sort of representation of the sequence of a gene, and well, what we did find was that two transcription factors cooperate on the promoter of CSPG4, and in the Duchenne, one of these transcription factors has a lowering of its activity, so the information is modified. If the two work together, they switch on the light. If one of the two no longer participates as co-regulator, the light is not switched on, but rather switched off. And this happens in DMD macrophages. And since all this mechanism, this promoter that we have been able to identify is placed inside an area that is affected by acetylation, so by histone deacetylases, we use givinostat that had already been introduced. It is a drug that is very well known for its anti-inflammatory, anti-fibrotic properties. It reduces the cytokine cascade, and it is being tested for DMD and other myeloproliferative pathologies. So we tried both young and old mice with givinostat, 20 milligrams, and then we checked what was happening to those animals. So we have looked at the tissue. This is the histology, and you see that givinostat modifies the mechanism, and so it restores the normal condition, restores an angiogenesis that is more similar to the normal condition, and reduces the inflammatory infiltrate and a higher presence of sympathetic innervation in the heart muscle. Later, of course, we have completed the heart analysis and proteomics and transcriptomics, and it has no effect on young mice, possibly because the pathology has is not present yet in the heart, whereas in older mice after givinostat, look at the color affinity, and the genetic profile is more similar to that of healthy animals. Then all the genes were rearranged. So you see the analysis has identified this protein that is clearly reproducible, and now it is being investigated by Dr. Berlucci. And all proteins and all the genes involved in the extracellular matrix are affected. Of course, the animals were examined with an ECG, and there was an increase in heart performance, and then a hemodynamic analysis as well. And here you have a massive reduction of the fibrosis in the tissue, and also a reduction of all the genes that are involved in collagen deposit. So we can conclude by saying that the macromolecule CSPG4 is highly expressed in the dystrophic cardiac extracellular matrix and is able to prevent proper cardiac innervation. Macrophages are the main producers of this proteoglycans, and the recovery of cardiac functions following treatment with givinostat is concomitant with the reduction of the expression of fibrosis and the remodeling of the proteins of the extracellular matrix. At present, these are the projects underway for DMD. We were funded for CAR-T CSPG on dystrophic pigs. We have a collaboration with a service, and we are carrying out the single cell characterization for patient samples and blood and bone marrow. And we are working on a 3D bioprinting that we have in our lab, and we are characterizing the inflammatory results to close this project with a machine that can do the screening. I wish to thank Marika Milan, who has been carrying out all these experiments for the last 5 years. Thank you.
Grazie infinite.
Thank you very much. Thank you. And now we seem to be running out of time. Therefore, let me introduce you to Professor Marina Boucher from La Sapienza University in Rome. She deals with forensics among the other issues. So thank you for being with us. You have the floor, Marina.
Ok, allora grazie.
So thank you again. This is about our project, which orbits around the identification of this enzyme, PKC theta, as a therapeutical target in DMD, namely DMD and cardiac complications. First of all, I wish to thank Roberto for dealing with basically most of my issues. So we are mainly talking about dilative cardiomyopathy. We know that there's an initial fibrosis, and then it may move on to becoming a real dilation. Now, guidelines seem to say that ACE inhibitors should be administered from the age of 10 years, but some patients are not eligible for this kind of a treatment, considering the use of corticosteroids that account for a good anti-inflammatory therapy, as we know, even though in the long run they may induce some side effects, whereby we need to plan some new strategies in order to modulate the immune response, possibly reducing toxicity and side effects. Of course, there may also be an association of the different drugs. Now, within this framework, a few years ago, we considered that by using the MDX mice, dystrophic mice, the absence of PKC theta, well, this could somehow modulate, even reduce the dystrophic phenotype. Basically, that happened in mainly very young animals, around 2 or 3 months of age. There was also a reduction in the myofibers necrosis, an improvement in muscle regeneration too. So here you can see. And the same goes hand in hand with preservation of physical exercise. So basically the wild type mice did exactly the same. But what about this PKC theta? Well, it is mainly expressed in the immune system T cells. It is a target for inflammatory diseases, and a very attractive one, because its activity is of paramount importance, indeed indispensable for the immune response to parasites or transplantations and so on and so forth, even though it is not necessary for an antiviral response. Hence, its attractiveness, since its inhibition is not supposed to disrupt the antiviral response. And This also goes for immunosuppressive T cells. So once again, it is supposed to have an immunomodulatory activity, which is good, of course. And this is precisely why it is considered as a possible therapeutical target for the treatment of several diseases, inflammatory bowel disease, asthma, and so on and so forth. Hence, our idea of including it also in the DMD possible treatment, namely by using a specific PKC theta inhibitor, the C20. Well, we could demonstrate that the treatment with this inhibitor leads to protection from the damage due to an excessive physical stress. And this goes hand in hand with the early inhibition of the PKC theta dependent infiltrate of T cells into the dystrophic muscle. So a very early treatment in very young animals before necrosis appears prevents such inflammatory infiltrate from appearing. It also prevents histopathological disruption and reduces the levels in serum CK. Hence, we concluded that the inhibition of such particular T cells may lead to an immunomodulation, so a reduction in inflammation, a reduction in necrosis, an increase in regeneration, which lead to an improvement in performance and in muscular integrity. Can this apply to possible prevention of dystrophic heart, so to speak? In other words, late accumulation of fibrotic tissue into the heart. Well, MDX mice were used, and then, of course, several aspects regarding the motor factor were considered. These animals already showed some heart impairments, but quite late, around the age of 11 or 12 months. And we wanted to understand whether within this window such a treatment could work. So here you see this late problem that arises at around 12 months of age. Then some hemopoietic cells have been used too, but the main heart problems are definitely less strong, weaker than in humans. So we tried to generate a model that could worsen the situation, and we used a protocol of moderate physical exercise, but in the long term. And the treatment lasted for some 2 months. So at 3 months of age, MDX mice were submitted to such exercise, which led to an accumulation of fibrotic tissue, which is more or less the same as the one we find in the heart of 11-month-old mice, differently from those that had not undergo any physical exercise. And we also get an increase in necrosis within the heart and also in the infiltration due to the use of hemopoietic cells, both lymphoid and myeloid. And we get a reduction in the heart function, in the cardiac function. And we have seen that also through the shortening fraction. So there's definitely a reduction in cardiac function in these 3-month-old mice submitted to such physical exercise. So treatment with C20 can definitely reduce inflammatory infiltrate in such mice. Here you see the histopathology that we have seen. So we can definitely get a prevention prevention here and a reduction in all the different immune response inhibitions. We also get a reduction in the size of the fibrotic tissue and the necrotic area too. And we also get a certain stabilization in the cardiac function as seen through the ECG. So in conclusion, we have generated an animal model through this, long-term but mild physical exercise protocol. We hope we'll be able to use this also at a later stage in order to better consider the accumulation of the fibrotic tissue, the increase in necrosis, and so on. So in any case, the pharmacological inhibition of the PCK theta-dependent immune response may prevent such heart impairments, disruptions. So we probably have to design new inhibitors and identify those populations that may actually be responsible for all inflammation-dependent fibrosis that may accumulate in the heart. So we need to study these populations more in detail in order to better detect the real and the right targets. And to conclude, let me acknowledge those who have cooperated with us. I'd particularly like to thank Jacopo and Biliana. They have really worked, side by side with us. I'd also like to thank those who have synthesized C20 and Professor Sciarretta for helping us carrying out the functional evaluations, as well as all our sponsors, but in particular the Duchenne Parent Project that has financed this particular study on the heart. Thank you very much. Thank you.
Grazie infinite, Marina, per il tuo intervento.
Thank you, Marina. And now I'd like to give the floor to Luca Tucciarone. We are slightly late, but never mind. And Luca is a young researcher, and thanks to Parent Project has developed his PhD project. He has been working together with Parent Project following our campaigns both over Christmas and over Easter. The project has been coordinated by Dr. Pier Lorenzo Puri and Dr. Consalvi of the Santa Lucia Foundation in Rome. The objective of this PhD project aimed at finding treatments that FAP can be modified and how they coordinate the cell activity so as to favor muscle regeneration in Duchenne. Thank you. It is a pleasure to be here, and during my PhD project, I studied inhibitors of histone deacetylase, HDAC inhibitors, and it is a set of molecules that inhibit HDAC and resume the expression of a number of genes that were depressed by the absence of dystrophin. And so we concentrated on this pharmacological approach, and we also carried out a preclinical study on the mouse model of DMD using givinostat that then went on to clinical trials. Now, many patients with DMD are being treated with givinostat with very encouraging results. But what does givinostat more specifically? In muscular dystrophy, in the absence of dystrophin, microdystrophin is not only a structural problem, so resistance of fibers, but the absence of dystrophin directly involves the absence of hydrosylation of HDAC. Inhibition of this enzyme in muscular dystrophin. Well, this is what happens. What do these inhibitors do? They correct this axis. They inhibit this enzyme and they recover the expression of all these genes. Surprisingly enough, in 2013, or rather when we started studying these inhibitors on muscle slices with the dystrophic mice treated with these inhibitors, this experiment was published in 2013, and they used TSA.
Una piccola cortesia, leggermente più piano, per favore.
Slow down, please. Well, the chairperson is asking the speaker to slow down. There is translation, maybe he doesn't know. We carried out some studies demonstrating that early stages of muscular dystrophy, dystrophic mice treated with these inhibitors show an increase in muscle regeneration, but also they prevent fibrotic tissue formation. This is early in the early stages, but in later stages, there is a resistance of the muscle determined by muscle regeneration due precisely to the fact that these are late stages. And so you don't have a strengthening of muscle regeneration, but still fibrotic degeneration is somehow slowed down. We know what are these cells, what is the cell population that is responsible for this fibrotic degeneration. These are the fibroadipogenic progenitors, this FAP. This specific cell population, even though it has no dystrophin, still it responds to these inhibitors. So my project studies the following: whether on FAPs we can measure and we can see if TSA and the inhibitors at large can reduce the enzymatic activity of various types of HDAC. And the first important discovery is that during the progression of the pathology, there is an important increase in the activity of these enzymes. But we've also seen that at the early stages of the pathology, TSA, are very good at reducing the levels of enzymatic activity. And this is something they still do at the late stages of the disease. But since they start at very high levels, they are unable to go as low as they go in the early stages. That was done in collaboration with the Moresi lab. What we are interested in is that, well, this is an experiment on enzymatic activity, but we are more interested in genic expression. HDAC are involved in the acetylation of promoters of histones. When acetylation is increased on the promoter of these genes, there is an increase in gene expression. When it is reduced, there is a reduction in gene expression. And HDAC reduce that by, do that, whereas HDAC inhibitors correct this axis, improving the gene expression and inhibiting this activator. Here we are at the very heart of the project. So we wondered how on the whole genome, but also more specifically, what were the changes in acetylation and what were the genes that were modulated. So this is the experiment. We took this from dystrophic mice, early and late stages, treated or non-treated with this inhibitor. And we did chip sequencing and RNA sequencing. Chip sequencing shows the acetylation on some of the regions of the regulatory regions of the genome, and whereas the RNA sequencing concentrates on the way they are expressed. So FAPs have a sort of cell senescence as the disease goes on. It is a sort of an early aging of the cells, and it has very well-defined aspects that are modulated by the acetylation on the FAPs, on the FAPs, during the progression of the disease. And this is linked to cell proliferation. And you have an increase in acetylation for elements that have pro-fibrotic and pro-inflammatory factors. They are called SASP, SASP. And you see here the effect of this HDAC in the various FAPs in the different stages of the disease. There are three great categories of biological processes that are being modulated: secretion, metabolism, and more specifically glycolysis, and the activation of the cell cycle, so the cell proliferation. Early stage FAPs respond to HDAC inhibitors, regulating all these factors, increasing glycolysis and increasing proliferation. Whereas late stage FAPs at late stages of the disease modify these factors in a different way. Both in terms of regeneration, there is a reduction in glycolysis and a reduction in proliferation. And here we are going into the detail. We wanted to understand how these biological processes were regulated, and FAPs at the latest stages of DMD release these factors that are associated with senescence, that are called SASP, and that are overexpressed. And with these, we see not only that there are factors that are detrimental to muscle regeneration, but also to extracellular vesicles that are essential in the communication between FAPs and the cells. These are progenitor cells of muscle fibers. And we have demonstrated that FAPs, when they come into contact with satellite cells, they potentiate muscle regeneration in terms of the differentiation of these cells. So we see here, and that confirms this data that already found in the literature, the communication between SAPs and satellites, is not really there, because there is a reduction in the differentiation of satellite cells when they come into contact with FAPs at the late stages of the disease. But we know that HDAC inhibitors strengthen this axis. We have recently published an article that explains how TSA modifies the content of these vesicles, increasing their content, and when they reach the satellite cells, they have this differentiation. And we see that TSA in the late stages of the disease inhibits any type of secretion of these cells, and the extracellular vesicles, the extracellular exosomes. TSA in this case, to avoid FAPs to degenerate more, it blocks secretion entirely. And these factors at an early stage were modulated in an appropriate way, and in a way that was useful also to muscle regeneration. These are the experiments that have verified the story I'm telling you. These are two experiments that check the effects of this HDAC inhibitors, early and late stage, glycolysis and cell proliferation. And this is the biological trade-off. We see that TSA in all FAPs switches off the signals, and some of them are useful if they are transiently activated, but they're all switched off, So in conclusion, what I've understood with my PhD during the progression of DMD, FAPs acquire some aspects of cell senescence. They are expression of this fibrotic genes, and there is a sort of blockade of the cell cycle. Then we have differentiated of these inhibitors in late and early stages. In early stages, they prevent they promote these pro-regenerative factors, they prevent the fibrosis, they activate the cell cycle, and they promote glycolysis, which is very interesting because one of the ways in which HDAC inhibitors in the early stage increase the acetylation of the genome is by increasing the glycolysis, because if you increase glycolysis, you increase the number of acetylic groups that are useful to increase acetylation, whereas in the late stages of the DMD, they block the expression of these factors that are being secreted, being them pro-inflammatory or pro-fibrotic or regenerative or whatever, whereas they are unable to reactivate the cell cycle or promote the glycolysis. That is why these inhibitors are unable to promote acetylation, as is the case in the early stages of the disease. This is my last slide. I wish to thank Parent Project for the support, for the possibility of working with them during the years. A special thanks to Silvia Consalvi and Pier Lorenzo Buri. And I wish to thank all my co-workers and thanks you all for the attention.
Grazie mille.
Thank you.
Luca.
Thank you, Luca. So now, before moving to the Q&A session, since, by the way, we are running a bit late, so please try and be brief in your replies, although keeping not too fast pace for the sake of simultaneous translation. So before going into that, let me introduce you to our project page restyling. Of course, you can have access to it through our site, or else, of course, you can use the QR code that is here in the slide. It's a brand new page. We often ask researchers to send us their video, and generally speaking, they comply with this request. Okay. So I think now the floor goes to Maria Rosaria Rollo for the Q&A session. We have three questions from Alberto Costantino and Angelo, therefore Professor Sorci and Dr. Chiappalupi. So first of all, what else do we need to reach to the human? And then what about the next step for clinical trials? And since sex have been tested for other diseases too, can we do without phase 1? So, good evening, everybody. First of all, I wish to thank you all for inviting us here and for supporting us. I would also like to thank the people who have asked questions, Angelo and Costantino in particular, since I know both of them, but the answer is not easy. So, first of all, the results we have achieved so far are very encouraging. Especially, I'm thinking of sex to be used in humans. They are certainly enriching the framework. You know, the sex are not immunosuppressive. This is of paramount importance. They do not stimulate the tumor growth, which is also very good. And then they have an indirect action on the muscle satellite cells, thus inducing its growth and their proliferation without, however, producing any myogenic loss. This is very important, of course. As for dystrophic stem cells, having sex in there will certainly, at the end of differentiation, produce some improvements. So these are very important data that we have acquired in order for us to move on to humans. So what do we need at this stage? Well, we basically need a company, you know. We basically need a company fully embracing our project and supporting it from all viewpoints. Right now, no testing is being carried out within the framework of other diseases. In other words, the MC sex have already been used. I mean, microcapsules have been used in humans, in diabetic patients in particular. As for sex, in their free nature, so not in their encapsulated nature, have been used in a Latin American test that has given rise to several ethical issues, I might add. Anyway, they were used together with the B cells of the Langerhans isle in the pancreas, again in diabetic patients. So they have been used all right, but of course with a different approach, with a different horizon, so to speak. There's a patent, by the way, regarding such MC sex, so we all seem to be willing for such a passage into humans to be the case. Will this be for DMD? We may not be sure about that. Actually, right now, we're not interested in that either. We want MC6 to be used in humans. Then, whether it is for the treatment of DMD or other diseases, we're not really interested in that right now. But all researchers seem to want this to happen, but of course we will welcome any suggestions regarding this. My answer to Danielle about the mechanism of action is the following. Now, this is quite well known since sex release some factors, some of which are not well known. Anyway, from the peritoneal cavity, they get into the circulation from which they are available to all the body muscles. Thank you very much, Guglielmo. Thank you. And now there is a question for Dr. Falzarano, again from one of our dads, so to speak. Any tests have been carried out on the urine of boys included in clinical trials, say with givinostat or exon skipping? Because it might be interesting for us to understand to what extent such markers are useful or not useful if the boy is on a trial. Well, this has so far not been the case. So boys in clinical trials have not been included in this kind of a study, which might be interesting, though, in order to see what this may change and what it may involve. Of course, we need to understand to what extent the therapeutical effect of this molecule is still there when the cells are then to be under culture, to undergo proliferation mechanism. So we don't really know whether the process will still be there once the boy is off treatment. And we also need to examine the differences between the pre and post treatment conditions. I think it will be a very interesting model to be studied. Thank you very much. And now there's a question again for Guglielmo's team and for Sara, maybe. And another one of our dads has this question. Which is the mechanism whereby sex injected into the peritoneal cavity can be used from a distance in a way? Well, actually, I'm sorry, Gloria, but I did answer this question. Okay, sorry again. I'm just reading all the questions, in a row. So now Maria Rosaria has the floor for other questions. This is from Costantino for Dr. Rizzi. How have you administered the Givinostat into mice? And were mice under cortisone, as it is the case within trials? Thank you for this question. Actually, we carried out an IP injection, no gavage. So that's this for Givinostat, and the dosage was 5 milligrams per kilo, much higher than the one normally used within trials. Well, this happened because we agreed with the Italfarmaco people, who also sponsored us, I might add, that massive doses of Givinostat should be administered in order to better detect the main mechanism. And then no cortisone, of course, because otherwise we would have dealt with yet another variable. Of course, in a clinical trial, all parameters should be controlled. Therefore, that has been our choice. We don't want to confuse the framework even more. Anyway, massive doses. These are the doses we used, basically 5 times higher than the ones normally administered within a clinical trial for a 1-year lapse of time, I might add, whereas we only moved within a 60-day lapse of time framework.
Grazie. Sempre una domanda per
Another question now
Dottor Rizzi.
for Dr. Rizzi.
Perdonatemi, in che modo avete somministrato Givinostat ai
How did you administer Givinostat to mice? Yes, so that was for the IP injection. I did answer that part of the question. And the same question from the same dad goes to Luca Tuccerone. So how did you administer Givinostat to mice? And were they under cortisone? Thank you very much. It's my pleasure to answer. We did not use Givinostat. We used DSA. We used it without cortisone and through an IP injection. Thank you very much.
Avevo una domanda sempre da un altro papà. Questa è abbastanza generica, chi mi vuole rispondere?
That's a general question. So for all those who wish to answer about BMD. So, is there any research about nonsense mutations? Anybody wishing to answer this question? Or maybe there's something in the pipeline regarding this. Maybe you want to move from DMD to BMD, some of you at least. Well, I am now inside the Policlinic of Milan, the Milan General Hospital. I know that phenotyping BMD and using givinostat in this condition is something that is definitely being considered now here. So I understand people are actually wish to move somehow from DMD to BMD. I know nothing more about this, but one thing that I do know is that the BMD study is definitely undergoing. Thank you very much. We'll certainly deal with BMD in the next few days. Maria Rosaria, any other questions you want to read out? Well, I have no more questions here. Good. I can still see one about it's for Dr. Rizzi again, and it's just in order to better understand what about the possibility of not having similar results in humans. The decrease in platelets detected in humans has never been considered, right?
Sì, allora fondamentalmente io ho un po' ristretto l'area sul cardiovascolare, per questo motivo.
I have mainly dealt with the cardiovascular area, so I didn't consider the whole vascular sector. So when the project started, Pier Lorenzo was, of course, behind my choice, because we were working in two neighboring labs. So he said, since you're an expert in heart conditions, why don't you consider this too? So that's what happened. I accepted, of course. I was very happy to accept that challenge. But actually, in the scientific field, we normally consider very small portions of investigation. You know, I couldn't have come up with any studies if I had had to consider the whole subject, at the same time. So we may move on to expanding on the subject later on. But actually, the platelet decrease that was mentioned, we did not consider it actually. Well, of course, then we moved from mice to bigger sized animals, and this might take us to interesting and promising results. We are currently working on porcine DMD porcine models, in which case the several physiological parameters that are involved are going to be tackled more in depth. But we did not do that in mice. I'm sure other scholars are currently investigating just that part of the story. Of course, we are here to study our specific sector. Thank you very much. There is one more question. It's actually the last question, because then Cristina will have the floor to tackle the issue of fundraising. But all the other questions that we will not have answered will be sent out to our speakers who will then reply. So one last question. And that is the following. What about the administration of mesenchymal stem cells from the umbilical cord? What do you think about this?
Non so, purtroppo il controllo del differenziamento.
Unfortunately,
a noi ancora non garantiamo 100% del differenziamento. Quindi
we cannot guarantee for a 100% stem cell differentiation, which means that it takes a much better structured strategy for us to use them. As for the mesenchymal stem cells, I really have no experience in using them for treating DMD. So quite frankly, I don't know, maybe Marina or Lorenzo, maybe Luca too, they may answer better than me. Well, actually, yes, I can take the floor to say that thank goodness we are leaving these, say, gross approaches behind. You know, yes, you have cells, you inject them and see what happens. Well, this is now behind our back because we are working at concepts. In other words, why do these cells have good effects? We believe that there may be an exosome secretory activity which might count. I mean, you don't set a problem in too general terms. So the same may apply to drugs. In other words, you start with a sort of a trailblazer, then you try and restrict the idea of the good effects, possibly also highlighting the negative effects. And the same goes with stem cells. We're trying to understand what the active principles possibly having a therapeutical effect are, ruling out the negative ones. You know, this is how it goes with cells. I mean, we've been working at them with this selective approach for years now. Thank you very much, Lorenzo. Of course, we'd love to talk longer with you, but maybe we'll see you again soon with other results. And by the way, cell therapy will be the object of some of our sessions. Thank you very much. We'd like to thank the interpreters, and I'd also like to thank Fernanda De Angelis and Ilaria Zito, who work with me. Thank you very much. Again, let me tell you, you'll receive the answers to all your questions. Thanks again. Thank you.
Ovviamente dovete rimanere un'altra mezz'oretta con noi.
Well, of course, stay for 30 minutes more.
We are drawing towards the conclusion of the first day. Please don't go away, because there are a few important things we'd like to share with you. I'd like to thank the rapporteurs of the former session and Gloria for chairing it, and all of you for participating. Thank you also for the beautiful parallel session we have listened to. And I have here Sara Ruggero, and we also have Roberto Zoccoli, who within the fundraising area follows the different events of fundraising. We have 1,500 patients with DMD and Becker. 800 families that gravitate around Parent Project Italy. We want to do something that is both necessary and useful. Gloria mentioned the huge sum we have been able to collect over the years. It's almost 4,500,000 euro just to fund scientific research, if we consider also the period before 2005. So this is the result. If each family were able to bring 1,000 euro of fund collection, or fundraising or small events. You know, collecting 1,000 euros is not really complicated. I mean, it is possible to do more. It is possible to do a lot. I mean, it is not asking for, you know, extra money or I don't know. I mean, funding research, raising money for research, funding the future and the quality of life for our children is the most important thing we can do. 1,000 euro per family, it is about 800,000 euro to be invested immediately, but we must be the first to believe in it. We must build a chain that links us to life and others, and certainly close to us will find people want to stay with us for this battle. We must ask help from our colleagues and friends and former classmates. We must tell about this disease, explain why research is important and why it is important also to support activities pertaining to life quality. Fabiana and Roberto will be sharing with us some of the possibilities we have. Fabiana, you have the floor. Thank you and good evening. I'm very glad this evening to have the possibility of sharing with you what is the commitment we have within the fundraising I mean, all the initiatives we have during the year. So immediately, I'd like to thank Elena, Irene, Aurora, Ida, Andrea, and Luke as well, and as well as Roberto is here with us and will be speaking about another small piece of fundraising. With these people, every day we try to develop all the activities that sometimes are organized very well, sometimes less so. We have a number of initiatives of fundraising that enable Parent Project to fund research, but also all that well, has to do with the quality of life of both DMD and BMD patients. Fundraising is one of the most beautiful and useful things to contribute to a change we absolutely want. We are the operational hand, so to speak, of this change, and we hope we can be a spur for our community because we try to organize events, organize fundraising to fund research and all Parent Project activities. But I mean, we must also involve clinicians, researchers, companies, citizens, not only families, all those who want to leave a mark in this world and a mark for our patient community and family community. I wish to thank all those who, despite the difficulties we have had in 2020, it was a complicated year for all of us, but many people actively participated in fundraising this year, and they believed that we would be able to achieve our goals. We have just closed the counting of the money collected over Christmas, then we have been able to collect more than 250,000 euro. We might have done better, but with all that was going on and the situation we were in, I mean, it was really a big achievement for all of us. It is just one of the achievements of this year, but we must move forward. We must continue with our activities. And that is why this morning we started by promoting on all our social channels. Possibly some of you have already seen it. And we have started, sorry, with our Easter campaign. And now, well, we are asking you to help us collecting funds for Parent Project and our mission. You can book products through Parent Project, contacting the association with all the addresses you can find on the internet website. And we ask you to get in touch with everybody, all the people you can get in touch with, neighbors, friends, friends, acquaintances, whoever. It is only by doing that that we can involve extra donors and reach the objectives we have set. Parent Project is us, it's all of us, it's the whole community rotating around patients with DMD and BMD. That is why each one of us is very important, really, to make the difference. This is the time when all of you, all the people who at the present are online, all the people will be contacted really to start this new adventure. So please support us, please support Parent Project, because thanks to Parent Project, each and every one of us moves on, so to speak, with his or her everyday life. The events are the time when we can tell a wider audience who we are, what we work on, and what our fight is about. Thank you, Maria Rosaria. And the Dystrophy Tour is an idea that, well, started 5 years ago, and it is the heart of the local events, of local sports events. It is a circuit. Well, there is a number of events of cycling events all over Italy. These events, of course, are aimed at fundraising for Parent Project, but they are also events where we can make our association known to a larger audience of sports enthusiasts. Over the years, the Dystrophy Tour has developed enormously, and also the number of places we go to has increased. And the characteristic of the Dystrophy Tour is the closing place. This is an event organized by Parent Project, and it is an event that is somehow a symbol of the work of 1 year. Initially, in order to organize with this closing moment, we sort of twined with other organizations, but then together with Lorenzo Bugliatti, we decided to do something different. We called it Dolomiti for Duchenne, and this is how this initiative started. It is something that is unique in Italy. It is a 4-day mountain bike event on the Dolomites. You see the closing picture of the last of this Dolomite event, where the participants, of course, can cycle in spectacular places, and they live community moments with the families of the association. It's something truly unique, because in Italy we have nothing similar, the passion for sport and solidarity. But if you want to participate to Dolomiti for Duchenne, you must have a pass, a virtual pass, and this virtual pass can be obtained by collecting a minimum of 500 euro for Parent Project. Then there are other steps for fund collection, higher steps, guaranteeing bikers to conquer this famous ethic sweatshirt. So over the year, during the year, a number of local events are organized by volunteers, by Parent Project families, and luckily, local events are not only linked to the Dolomiti for Duchenne or the Dystrophy Tour. We are speaking of cooking events, concerts, exhibitions, or even different forms of festivals where the local tourism organization decide to give some of the money collected to the organization. Organizing an event is very easy. Well, the essential ingredient is passion and the decision to organize something beautiful. As Parent Project, we make all our skills available, graphics, logistics, and all that is behind the organization.
Ne sono stati fatti tantissimi, di grandi, come ad esempio la cena di Bergamo nel periodo natalizio, lo spettacolo di Natale organizzato da Gianni Bontempi e tanti altri esempi. E quindi
many other examples, of course. So it's good to see the extent to which several groups get closer to our association, choosing to spend their time in order to raise funds. Let me give you another example, the example of San Bellino, a very small town. Well, the number of participants there has outnumbered the population, the local population. But I mean, I may give you many more such examples. Anyway, such events are of paramount importance for fundraising, as we have said, but also for another reason, because people fall in love with Parent Project, because they get to know the families, they get to know their stories, the story of the boys, of their families, including their siblings, which links us much closer. So we are firm believers in our events, mainly for this reason. And again, this is an example of how possible it is for people from all around Italy to get together, once in a year. But even so, they seem to have been long-time friends because they share the same objective. So please come to see us in our Facebook page, but also on our website. And of course, we always try to update this, also making the people who are interested better and better aware of our whereabouts. So thank you again. Hopefully I'll meet you in Villa Bassa soon for the event that will be held there from next June, from the 18th to the 20th. Thanks again. Really, I wish to thank all volunteers because they help us and support us. Thank you very much. Thank you, Fabiana. Thanks, Roberto. And thank you all. Thank you. I'm sure you will take an active part in our next Eastern campaign. This will make you happy, believe me. And tomorrow we'll meet again at 4:45. Thank you very much. See you tomorrow.
Grazie, buona
Buona
serata.
Ciao a tutti.
Buona serata, arrivederci.
Grazie infinite.